This Week in Microbiology is brought to you by the American Society for Microbiology at asm.org/TWiM. This is TWiM, This Week in Microbiology, episode number 301, recorded on December 28th, 2023. I'm Vincent Racaniello, and you're listening to the podcast that explores unseen life on Earth. Joining me today from St. Louis, Missouri, Petra Levin.
Happy to be here.
Hello.
Happy holidays. Happy almost New Year.
This is our last TWiM of 2023. We got to 301. It'll probably be released next year, though, I'm guessing.
So Happy New Year, then. Happy New Year, everybody.
Yeah, Happy New Year, everybody. In 2024, another year of TWiM microbiology. If you enjoy our work here on TWiM and all the other podcasts that we do on Microbe TV, we'd love your support. Normally, I would say go to microbe.tv/contribute. But for the month of January, Daniel Griffin over at parasiteswithoutborders.com is doing a fundraiser for Microbe TV. He has a 501(c) nonprofit. Every few months he supports various causes, and for November, December, January, it's Microbe TV. And the cool thing is, whatever you donate, he will match, so you get double your money.
Those are the best. Those are the best for matching.
Yes, matching. So at least for January, go to parasiteswithoutborders.com. And then after that, you can go to microbe.tv/contribute. And we always appreciate your support. Had a visitor today at the studio, Petra. From time to time, people want to visit. And this woman is an animator for The Simpsons. Which is not something I watch, but I've heard of it, right?
It's awesome. We used to watch it all the time in grad school.
She brought me a T-shirt and a cap and a poster with, what was the girl's name? The young girl. Do you know her name?
Kathleen.
Lisa.
The baby is Maggie, and then there's Lisa, and then there's Bart.
So Lisa, according to Sylvia, is the lady who's the animator, and she's a fan of our programs. Lisa is the most science aware of the whole family. And so she has a bubble over Lisa on the poster saying, you are my best teacher, or something like that.
That's really nice. My favorite is when Lisa tries to teach Bart to be more contemplative, and she asks him what the sound of one hand clapping is. And he basically tries to slap his hand together, just one hand. He's like, easy, Lise. And so she's always trying to get him to think more deeply, her older brother.
Yeah. Anyway, that was fun. I learned all about animation too, which I knew very little about. All right, but back to the topics today. We have two papers for you, and I'm going to do a snippet which has to do with bacteriophages. It's in Nature Microbiology. It's called Phage Diversity in Cell-Free DNA Identifies Bacterial Pathogens in Human Sepsis Cases. And the first, I could read all the authors, but there are two co-first authors, Naomi Haddock and Leila Barkal. Then we have Nikhil Ram Mohan, Gurno Kaber, Charles Chu, Ami Bat, Samuel Yang, and the corresponding author is Paul Balicki, and the groups are from Stanford University, which is, of course, in Stanford, California. So this is a completely computational biology paper, which is fine. It's not to disparage it in any way. There's just no wet experiments, I should say. But what they're trying to do is understand what's happening in sepsis, right? So sepsis is a condition where you have typically bacterial infection and rampant inflammation accompanying it. The goal is to try and get better at diagnosing sepsis rapidly, not take time, because the patients are often in critical condition. So we mostly use culture methods, right? But there can be problems with those. And so now the idea is, can we use next generation sequencing or deep sequencing? And what they are looking at here is what's called cell-free DNA. We have an acronym for it, cfDNA.
I didn't know there was an acronym.
Yeah, I didn't know either. These are small DNA fragments, 50 to 200 base pairs in length, found in your circulation and in your blood. If we took your blood, we would find lots of these small pieces of DNA. DNA is everywhere. It's mostly human DNA in your blood, but there's also microbial DNA as well. So, next time you think about getting injected with a vaccine that has a nucleic acid component and you're worried about it, forget it, folks. You're full of DNA already. It's not going to be a problem. And you eat lots of DNA as well. So anyway, the point here in this paper is to see if they can use phages to maybe identify the cause of the sepsis, right? Because phages are everywhere in our human body. Did you know that? They're everywhere. Every compartment.
All the bacteria in our body have phage, so then they're going to be everywhere.
Bacteria are everywhere. So are the phages. Even on your skin, in your brain, in your gut, your respiratory tract, your fingernails, everywhere. So, and the cool thing about phages is that many of them have a very specific host range, right? So maybe you could take advantage of that to look at and say, could we look at phages in this small DNA in the blood and say what bacteria is causing your sepsis? So that's the idea here. And so what they have done is used plasma samples from 2 independent studies. And these are patients with sepsis caused by infection. Sepsis can be caused by other things as well. But these are infection-driven sepsis. And then they have, of course, healthy controls. And then they do genome sequencing and try and figure out if they could use the phages to identify what's causing the sepsis. I mean, in the end, you can – it's not ready yet. It's not ready for diagnosis, but there's some interesting observations that they make. And in fact, they say, people are trying to use this cfDNA, bacterial cfDNA, to do diagnosis. It's very hard. Apparently, it's not working very well. And the phage may be better. So here's what they did. They have a Stanford study where they had 61 patients with sepsis. These are all patients seen in the emergency department. And then 10 asymptomatic controls. The sepsis are from 20 different bacterial infections, which they can do by doing blood cultures and saying what's growing in the blood of these patients. And so
So how long does a blood culture take? I think that's part of the reason they want to do this is the reason to do this isn't that we can't identify them. It's that it takes, I think, 24 to 48 hours. Do you know what
that's right. I don't know. Michael Schmidt would certainly know. But Michael Schmidt's in the air right now.
I know that when they sped up how quickly they can push things through at the lab at our university hospital, that it actually threw off the timing on the doctor's side because they had a certain routine that they would drop off the samples and then a certain shift would pick them up with the diagnostic. But then when they even changing it 6 hours made a difference.
Yeah, 1 to 3 days apparently.
Yeah. So that's why you want to do this. You want to be able to sequence because then you can get the results back without culture. It's super fast. And then you know what the bug is, which is definitely a big step in the right direction.
Yeah, otherwise you have to use a general antimicrobial, right? And
you can use a specific one if you know it's, E. coli or Klebsiella.
So they collect the CF, cell-free DNA, and they sequence it and get a lot of DNA sequence. And then they do a lot of computational manipulations of these data. They have to remove all the human sequences, right? Because there's a lot of that. And they have a lot of programs with interesting names. I like the one program called Bowtie 2. That's a cute name. What I found interesting is that less than 1% of the reads correspond to mammalian viruses. So in the blood, there are nucleic acids of mammalian viruses, even in the healthy controls. All the samples would have that. But about 8 to 9% of the reads were bacteriophage specific.
Actually, that doesn't surprise me that they're so low because the bacteria numbers are so high. And if all the bacteria have viruses, then that's just going to amplify the numbers. It also doesn't surprise me that there are human viruses in there because we are infected all the time. There's a nice study actually out of some pediatric study where they show like kids who come into ER with like broken legs have human viruses, even if they don't have any particular symptoms.
Yeah, that's right.
So how long again are these reeds? They're really short, right?
Yeah, the Illumina reeds are short. Yeah. That's part of the problem. It's sometimes hard to align them and find out what they're from, right?
Right. If you don't have enough length of sequence, your probability of getting a unique sequence is low.
I mean, they can certainly do longer reads, like those little cartridges, the Minion cartridges. Have you seen those?
I have the MySeqs and they're very impressive.
It's a little cartridge about the size of a harmonica, and you put a drop of your sample in, you close it, and then you plug it into a USB port on a computer, and it just generates sequence. And I was in Germany a couple of weeks ago, and I went in a lab, and there was one sitting there, and it's just amazing. And this is going to give you long reads, millions and millions of reads, and you can mix samples. You can have hundreds of different samples all mixed because they're each barcoded. It's just amazing, the technology. Anyway, yes, 8 to 9% phage, and all the samples had phages. Remember, they have an asymptomatic cohort here, and those also had phages as well. So the problem now is, can you link the phages with the bacterial host, right? And they try a lot of different things, but in the end, they say there is no broad sepsis phageome signature, right? There's just nothing that screams sepsis. All right. So then they have to do some manipulation. All right. So what they do is they say, okay, sepsis is usually, not always, but usually caused by a single bacterial strain. And it can be E. coli, streptococcus, staphylococcus, Klebsiella. So they said, can we look for phages specific to those bacteria in the data sets? And so the answer is yes, you can find an increase. So there's this metric called the Shannon Diversity Index, which is a statistical measure of the diversity of an organism based on genomes. And so you can use that to look at the diversity. And they find that the Shannon Diversity Index is higher in patients with, say, E. coli, strep, staph, Klebsiella. They can find phages that are specific to those bacteria. All right, but you can't really read the phage sequences and say this person has one of those bacteria. That's the problem.
Right. So if you cultured them, though, you would get that one bacteria out.
You would. But again, that takes 1 to 3 days. And so.
Yeah, no, exactly. So it's like a tension between time versus.
Yeah.
At this point, though, they're probably treating the people anyways. They'll just give them a broad spectrum because sepsis basically sends your body into shock.
Right.
And you lose blood pressure and you cut off blood to your organs and you start to die. So you have to treat it really rapidly and very aggressively.
Yeah.
No matter what.
Yeah, so they do go on broad spectrum until they get a report back from the clinical lab, and then they switch. Yeah. So they then repeated this study with a larger group of patients. This is 391 people. There are 4 different populations. They have blood culture positive sepsis. They have blood culture negative sepsis with known infection. They have what's called systemic inflammatory response syndrome. These are people who have sepsis. They have a lot of inflammation, but it's not infection driven. And then they have asymptomatic controls. And again, using their DNA sequencing approach, they find that, again, the phageomes. So a phagome is a word for all of these sequences from the blood in this case. They're similar in both health and in sepsis, right? And so that's one thing. And they, again, could, if they knew the bacteria infecting, which they do for many of these cases, they can match phages to it. But again, they don't find a sepsis phagome, even in this bigger cohort of patients. Now, the next step they do is that E. coli are often common causes of sepsis, and they look for E. coli phages. They say they're sporadic in asymptomatic patients. So some have E. coli phages, but not all. But ubiquitous in people with E. coli sepsis. So everybody with E. coli sepsis has E. coli phage, which is not surprising, right?
No, that's good. E. coli are a very small portion of your microbiome, so you would expect very little E. coli phage in your blood unless you are infected. So that's actually a good way to go. This is a nice breakdown. Like, they try one thing, they have to get narrower and narrower and try and figure out, kind of hone in on what they want to do. I like this.
As they say there are a lot of different E. coli phages, over 500 unique phages in 36 patients, and they could put them in groups. And they say we could find a group of phages pretty much present in all people with E. coli sepsis, but not in other groups. So, again, they're narrowing it down a little bit. And in particular, this is interesting. They have a group of people with sepsis that have toxin-producing E. coli, right? So not all E. coli are producing toxins. And these have specific phages associated with them as well. There's also a group of patients that have staphylococcal sepsis, right? And so they say, can we leverage this in some way? So Staph aureus in blood is always a bad sign. Right?
Never good.
Never good. Whereas if you have coagulase negative different staphylococci, that's often considered a contaminant. So that's not a problem. But Staph aureus itself, big problem if you have that in your blood. They can see that, sure, people with Staph aureus sepsis have phages that are distinct with people that have just these contaminants, these coagulase negative staph. And so you can distinguish them in that way. So again, increased diversity of Staph aureus phages is specific for Staph aureus infection. So that's basically what they find here. There's a little bit of, so everybody has a phagome, everybody. It's reflective of the bacteria that are in you. And you know what's interesting, Petra? They have a number of gut phages that are in the blood, which means that they're traversing, right?
Oh, right. Well, yeah, I guess it does. I'm wondering, though, in a way, like the phagome, when I first saw this, I thought this was really a cool way to look for things. But there are definitely unique sequences in the bacteria. Like E. coli has unique sequences. It seems more targeted to look for E. coli specific signatures. But I was then thinking, well, the phage are going to be amplified, so you're going to see a lot more. So I don't know. It's kind of, it would have been nice to kind of have some, I mean, there must be a review article out there. Maybe it needs to be written, and that comparing different ways to use sequencing to identify infection, because obviously, if you can sequence quickly, which still, I think people we're still using mass spec to identify pathogens, but like, if you can sequence, why not sequence a signature for E. coli, or even you could have one specific for urinary pathogenic E. coli, which is a common cause of sepsis in catheterized patients. So I thought it was really an interesting idea, but I'm not sure it's the most, I don't know. It's really interesting to think about.
I think that they say we have to look at more patients and see if we can, develop a better way to identify. But at least there's some, when you have an E. coli sepsis, there's an overabundance of E. coli phages. So, but when you just take the whole population, you don't see that, right? Unless you look for the E. coli specific phages. So there has to be a way to deal with
this
that.
Feels also like a great potential for AI to take some of these kind of big data sets and see if it can kind of figure out how to do it.
Exactly right. Exactly. So this is really a first pass here, but it has potential, and I think we'll probably see more of it, right?
Yeah, I think so. Well, also, I think it would be great to use phage as a way of looking at microbiome diversity, because they'll be specific. I don't know what the markers are, maybe changes in that, and then compare that to what the bacteria are, can tell you about the health. I don't know. There's all sorts of potential to use sequencing in new ways, especially now that it's fast. And as you said, you can get these little, basically a thumb drive sequencer, which is amazing.
All right. So that's that. All right, Petra, what do you have for us?
So today I'm talking actually about something I find really interesting. We talk a lot about antibiotic resistance and in this program and in general. And these are just genes that bacteria carry that essentially allow them to grow in the presence of antibiotics. They're different kinds. These genes encode different enzymes usually that either destroy the antibiotic through a chemical change. So beta lactamase is a classic one, or they actually compete with it. Sometimes there are changes in transport of the antibiotics, but they're usually often carried on genes that can be carried on either mobile elements or often on plasmids. So these are the little circular pieces of DNA that can replicate kind of independent of the bacterial chromosome, and many of them can be transferred between bacteria. The problem with plasmids as a transfer means in general has been, so you can have a plasmid that carries an antibiotic resistance cassette, and the idea is that another bacteria that isn't resistant to that antibiotic might pick up that plasmid and become resistant. Plasmids, though, typically have a limited host range, meaning an E. coli plasmid doesn't replicate very well in other bacteria, even some of its closer relatives.
What's the reason for that? Is it an origin of replication issue?
It's usually, it's not completely clear. It's usually a combination of you need some proteins that are on the host chromosome, which this paper gets into, to essentially ensure that plasmid is maintained, as well as things on the plasmid. Sometimes it has to do with sharing of replication proteins that aren't conserved between organisms. And it depends on the plasmid. We have E. coli plasmids that replicate very well in Klebsiella, which is a close relative, but would not replicate in Bacillus subtilis, which is a very, very different bacteria from a totally different group, evolutionarily super distinct. And the other thing I always find interesting is we talk about antibiotic resistance, and it's definitely, a problem, sort of like evolution in action here, because we treat people with antibiotics. When you have an infection, you take an antibiotic, and that selects for bacteria that have these antibiotic resistance genes in them, and the other bacteria are killed. And so you're sort of enriching that population over and over again. But we forget that plasmids and even antibiotic resistance cassettes existed way before 1940, when we really started to ramp up, 1945, when we started to ramp up the use of antibiotics. And so one of the questions is, why were these genes around, these antibiotic resistance cassettes? But another thing is, how were these plasmids maintained in the absence of selection, right? If you're not treating bacteria with drugs that select for the plasmids, how do these plasmids stay? And again, they're very common even in environmental isolates that, have never seen a patient or an antibiotic. And part of that is probably competition. Back in the soil, they're seeing that bacteria are interacting with other bacteria or fungi that secrete antibiotics. But again, there's a lot of plasmids that are very able to they're very stable and don't require a lot of, for any selection really, for many, many, many generations to remain. And this article, which is entitled Evolution, Persistence, and Host Adaptation of a gonococcal AMR, antimicrobial resistance plasmid, that emerged in the pre-antibiotic era, I think kind of addresses some of these issues in a really nice way. It's in PLOS Genetics, and it's by Yi, Yasser, Turner, Baker, Sehoven, and Tang. And they're all at the Sir William Dunn School of Pathology at Oxford in the UK, and Quadrum Institute in Norwich, also in the UK. So again, they're looking in Neisseria gonorrhea, which is the cause of gonorrhea, which is a sexually transmitted disease. It is easily spread. It's one of the most common ones out there. It's pretty much asymptomatic in many or most people. It can cause some pain, and in women, it's associated with infertility if left untreated. It's actually one of the interesting history of it is it was one of the first bacteria to be treated with penicillin during World War II. And there were lots of debates because there were a lot of sexually transmitted diseases among soldiers. One of the big debates was whether we should use precious penicillin supplies to treat an STD. But in the end, you want your soldiers to be healthy so they can fight, especially in the Allied side. So they made the decision to use penicillin to treat gonorrhea. After World War II, they started giving penicillin and other antibiotics prophylactically to people who didn't necessarily have it, but to prevent them from getting it. And this led to the selection very quickly of resistant strains of gonorrhea. And they've been through several classes of drugs, most recently the tetracycline and that group of drugs, which are protein synthesis inhibitors, which were discontinued in the UK in 2004 and in the US in 2017 because all the strains of gonorrhea that they were seeing were resistant. So there's no point using that drug. So this is looking at actually a plasmid, so a little circular piece of DNA in Neisseria gonorrhea that carries tetracycline resistance. And they are really just looking at it in part, and they're answering some questions. Why this plasmid has a pretty limited host range. It's really only in several Neisseria species. It's not even across the the genus Neisseria. It's not even all the species. And how is it that they're limited to specific hosts? Because again, plasmids, many E. coli plasmids will replicate in Klebsiella or Salmonella. So why is this particular one so picky about its host? And the other question is, how are they maintained in the absence of selection? Are they maintained in the absence of selection? So this plasmid is called P-Con-J or Con-J, and they track it, the evolution of it in this paper, and they look at its impact on fitness. Sometimes plasmids, why lose a plasmid? Because you're trying to bacteria trying to replicate this extra piece of DNA. It's easier if they don't have it. So there is a small fitness cost usually about having these little plasmids. And so the first thing that they do is they look at the plasmid to try and see how it might be maintained. And they see that it has partition proteins called par A and par B. And these proteins really help ensure that the plasmid replicates each time the bacteria replicates and gets separated into the daughter cells. It's usually not enough to maintain a plasmid, but it's one way. They also see encoded on the region that they call the gene load region, which is outside the replication area. They see two what we call toxin antitoxin systems. So these are kind of two proteins. One is a toxin that can kill usually other bacteria or potentially gonorrhea, and a second one that's the antitoxin. And if you just have the toxin, that kills the cell. But if you have the antitoxin, that protects the cell. So you need them together. They also see on this, so they have two toxin antitoxin systems, and then they have a third protein that is on there called VAP-D. And VAP-D is actually only one half of a toxin antitoxin system. It's the toxin part. So it has the two full TA systems, and then basically half a TA system. And they speculate that maybe that's why it's conserved, is it has these toxin antitoxin systems in par D. And so they, in the par A, par B systems. And in figure 1, you can see a picture of the sequence of the plasmid with par A and par B. The Ds are marked where tetracycline resistance is, the tetracycline resistance cassette, which is usually a pump that pumps TET out of the cell so it doesn't do any harm. And then, they've labeled all the things. And one of, I think, the cool things in this plasmid, they show sort of some evolutionary work to show that this plasmid appeared in the clinic over 80 years ago before really we had selected, certainly before we'd selected for tetracycline resistance. And it's in multiple lineages of Neisseria gonorrhea. And that says that this plasmid has been there, it's carried the resistance cassette long before we were using a drug that would select for it. So it's really well maintained, even without selection. And it makes sense that gonorrhea rapidly became tetracycline resistance when you're using TET as your primary means of combating it, because this plasmid was already there in many, many strains, and you're just going to kill all the strains that don't have it and end up with primarily strains that carry this pCon-J. But again, the bacteria had it long before it ever saw tetracycline. The next thing they do is, this is amazing, this plasmid's carried, it doesn't seem to impact fitness. They want to know what's required. So they do something which I think is a really nice technique called transposon sequencing, or TN-seq. And basically the idea here is you have a little piece of juncoming DNA and you jump it, they do it into the plasmid and into the chromosome, and they look for things that are only there, like you only see them when you're not selecting, essentially things that are required for growth under selection. And that means that essentially to keep the plasmid, you're looking for things that the cells will lose the plasmid when you don't have selection. So if you're not giving them tetracycline, if you make a mutation in a gene required for the cell to keep that plasmid, then that transposon that makes that mutation, you'll only see it when you don't have when you have selection, but not when you don't. Sorry to butcher that. But basically, it's a nice way of finding things required for selection. So they do this and they don't find any single gene required either on the plasmid or in the chromosome. And so their conclusion for that, and we'll get back to this, is that it requires multiple genes essentially working in concert. And so that if you knock out one, it's still pretty good at surviving on the plasmid, or if you knock out one on the chromosome, it's still pretty good at maintaining the plasmid replicating and staying. So essentially it takes a bunch of genes, probably both on the chromosome and on the plasmid, is the conclusion. They also show that pCon-J doesn't impact the fitness of the cells of the bacterium. So again, making plasmid is kind of problematic. In figure 2, they show that plasmid is prevalent. pCon-J is prevalent even in, they take strains from 2010 when we were using tetracycline selection, and they look at strains that were sampled between 2010 and 2019. So that's 2 years without tetracycline selection in the US, and between 2007 and 2016 in the UK. And so that's 12 years without tetracycline selection. And you can see that in figure 2 that half to a third of all the strains, in many cases, even as late as 2019 in the US, still have tetracycline resistance, even though they haven't been selected for. So it just reinforces that whatever system these plasmids contain to maintain themselves, so they're passed from mother to daughter, are really good, and that just losing selection doesn't make any difference. It's actually a really nice figure. It's a kind of fun experiment.
But you would expect randomly things get lost, right?
Right. If there's a fitness cost, so over decades, if there's any kind of fitness cost, like
sure.
if it slows the growth of a bacteria and the bacteria that don't have the plasmid grow faster, then you would expect to quickly have the population overtaken by the ones without the plasmid. But that's clearly not happening. So that says there's not really a fitness cost, at least one that makes any difference over decades.
Yeah.
So that also reinforces like these plasmids were there before selection and they weren't really making much difference.
So if there is no fitness cost, then you would expect it to stay forever. There's no...
Right. You would expect it to essentially be maintained in the population if it makes no difference.
Because the plasmid encodes things to make sure it stays for the plasmid's benefit, right?
Exactly. You can think of the plasmid like, sometimes we say selfish DNA, but basically the plasmid is really just trying to stick with the bacteria and be as quiet as possible. So the PAR system and these TA systems, they hypothesize, are really there so the plasmid can stick around.
Yeah.
Which is actually very impressive, but also a little scary because most bacterial pathogens have 1, 2, 3 plasmids in them. That's just pretty common, just like we were talking about some phage. Most bacteria are infected, sometimes stably with a phage. Plasmids are similar in that regard.
Right.
So again, pCon-J, their TN-seq data didn't suggest one thing made any difference. So then they systematically go in and they just make mutations in the individual TA systems, in PAR, and in VAP-D, this toxin. So again, the toxin shouldn't be there without something else. So that's a little curious because again, the toxin would kill the cell. So it suggests there's something probably on the chromosome that's protective against the toxin, a chromosomally encoded antitoxin, if you will. And in figure 4, they go through and they look at loss of plasmid loss. So they take the plasmids that are missing either the entire region that contains the two toxin antitoxin and the TWiM and the VAP-D toxin, or those three two full TA and half TA toxin antitoxin systems, and they look for loss. And they actually see if they knock out them individually, they don't really see much difference it's only when they knock out that whole region of the plasmid. And even then, after I think 40 generations, only 90% of the cells still retain that plasmid. That's knocking out, this big region. And they see it's actually really amazing. So 90% over 40 generations still retain it. So it's only when they start to combine them even that they see 10% gone. Individually, they really see almost no detectable difference. It's below the limit of detection, for example, when they knock out one of the toxin antitoxin systems. So that's, again, very impressive. This plasmid is really designed to stick with Neisseria. So again, they still have this problem. They have a toxin on a plasmid that's not killing the cell. And they don't understand how it's contributing to plasmid maintenance. It makes perfect sense if there's some other plasmid or part of a chromosome that has that antitoxin, because then the two have to stay together, right? You can't have the toxin without the antitoxin because the cells die. They actually show that if they express VAP-D, this toxin in E. coli, survival is 2 logs down. So clearly VAP-D is a toxin in some way. So to really understand how that works, they started to look in genome sequence data to look for cognate. So VAP-D has homologs in Haemophilus and Helicobacter, 2 other human pathogens. And those organisms, it's usually with the antitoxin VAP-X. So they look in the genome sequence data from Neisseria gonorrhea, and they look and they find a VAP-X homolog on another plasmid, which is called P-CRYP or PCRYP. And it turns out that mostly when they go through different isolates of gonorrhea, they see P-CRYP co-occurring with P-Con-J. So these 2 plasmids come together, or almost always together. And that, again, kind of doesn't completely perfectly confirm their hypothesis that they're working together, but it does explain how you can have VAP-X and the cells survive, whereas VAP-X alone will kill E. coli. And I thought this was really nice because they're going step by step and really showing it. What they do in the end is they go back to this idea that it's interplay between the TA, the toxin, antitoxin, and the partitioning systems that's really required. And they have a nice model in figure 6, and they show the multiple mechanisms that allow P-Con-J to be maintained. First of all, it has the 2 classic toxin antitoxin systems, and you need those together so you have equal amounts of toxin and antitoxin at all times. You also have this extra plasmid called P-CRYP that expresses the antitoxin for VAP-D, which is on the P-Con-J plasmid. So it shows it working really nicely. And as a kind of final thing, they treat the cells with fluoroquinolone. And fluoroquinolones and ciprofloxacin, which is a common antibiotic, we often call it just Cipro, it targets DNA gyrase. And that would be a common drug used now to treat gonorrhea. It's a broad spectrum. Vincent was just talking about broad spectrum antibiotics. Cipro is a classic, kills almost all bacteria. Cipro targets DNA gyrase. DNA gyrase unwinds supercoils. So if you take if a string, right, that has two strands and you pull on those strands, you essentially force the twists in those strands down and you get like coils of coils, twists upon twists. And that's bad if you're replicating your DNA, which is really two strands. And so gyrase makes sure to relax those supercoils so that the replication machinery can copy the DNA. So if you treat cells with ciprofloxacin, you end up with all this supercoils, DNA breaks, and you kill the cell. And they found that if they treat cells with Cipro, that eliminates p-Con-J. Even low levels of Cipro will eliminate this plasmid. So after 48 hours in the presence of kind of low levels of ciprofloxacin, 7.6 of the cells no longer had the plasmid, even though plasmid really has nothing to do with resistance to fluoroquinolones. If they treat the cells and they do a nice control with spectinomycin, which is a protein synthesis inhibitor, they get only 1.6% lose Con-J. So essentially they're showing they can get rid of the plasmid by treating with ciprofloxacin, and then they nicely go back to sort of clinical literature, which shows that the TET resistance frequency in patients who've been treated with Cipro, if you treat gonorrhea with ciprofloxacin or another fluoroquinolone, this patient's not only the gonorrhea that's left tends to be sensitive to TET, probably because they've lost p-Con-J. And I thought this was really nice because often we treat and we see certain things, but they're really going into the molecular biology of how this plasmid is maintained, why the cells retain TET resistance even after we stop treating with TET, which is unusual. And then also why, treating with one antibiotic might essentially result in sensitivity to a completely different class of antibiotics. So I really liked it. It's got a lot of nice techniques in it too.
So normally, you would say normally most plasmids impose some fitness cost, and that's why we see they're lost, right? So this is unusual in that it doesn't. Would you say?
I would say some plasmids, especially like the high copy plasmids. So those of you who've worked, who are undergraduates or taken an undergraduate microbiology lab, you probably work with plasmids that are ampicillin resistant. Often those are puck plasmids, or maybe you've used a pet vector. Those are high copy plasmids, and they won't be maintained if you don't maintain selection. But there are actually quite a few plasmids out there. They're usually very low copy plasmids. A good example is E. coli has a little plasmid called F, or the F episome. And that plasmid actually can transfer really easily between E. coli strains. It actually builds a little channel when the strains are really close together, and it can go between them. And that plasmid is really well maintained in the absence of selection. And many bacteria have plasmids without selection, again, if you pull an environmental sample. It was just interesting here because gonorrhea is a big problem. It's, again, a very common STD. It's easy to treat if they're sensitive to the drug that you're using to treat them with. And people hadn't really, I think, thought about them having a plasmid that gives them TET resistance, but clearly they do. And clearly it really argues against using TET as a drug to treat it. And for now, Cipro works, I think so.
I mean, I remember the days when I grew plasmids just to make DNA, right? Viral DNA. And we have to always have selection. Otherwise, plasmids would go immediately, right? Because they're high copy number.
High copy number plasmids. So these are really low copy plasmids. And again, even the low copy plasmid that you might use in lab, like we sometimes use a low copy plasmid if we want to put if we want to put, say, a puck plasmid or a pet vector in a cell and we want to have another plasmid in that same cell, we use a different origin of replication, and that usually is a low copy. And usually we still need two drugs to make sure we have one on the low copy plasmid and one on the high copy plasmid. And those plasmids, like a pet vector, has a big fitness cost because the cell's making, hundreds and hundreds of copies of that plasmid instead of using its, nucleotides to make more DNA. And that has a big fitness cost. So that's the other reason those plasmids are lost.
We wanted just as much plasmid DNA as we could get, right? So
we were just abusing E. coli for our own purposes.
With no appreciation for the biology behind it, right?
Yeah. Exactly.
It was a tool. We would grow a liter overnight ampicillin, and then you get all the plasmid you want. You didn't care. And sometimes we would have genes that were toxic, and you couldn't even grow them in a high copy, so you had to use a low copy just to be able to grow them.
Right, and then it's very frustrating because you get hardly any DNA out.
Exactly right. But, some of the first cloning vectors were low copy. The famous one is PACYC. Came out of Stanford. Annie C.Y. Chang, right? She developed it. ACYC.
Oh, that's where PACYC. We still use PACYC as our companion vector. When we have a toxic gene, we have a protein we often clone through E. coli that's a bacillus protein. And in order to get them to grow really well, we have to have something else on a PACYC vector to keep E. coli happy.
Yeah, it's we dreaded having to use PACYC. It brings back such memories. But the most striking part to me is that we had no appreciation for the biology. We just needed as much as we could get because we were using it as a tool, right? But here, this paper shows that there's a lot of underlying subtlety that's very, very cool when you figure it out.
There's some amazing plasmid biology. There's some really great work from Joe Poliano's lab where there are plasmids that have special partitioning systems that use cytoskeletal proteins to push the two plasmids to the poles of the cell. So then when the cell divides, each daughter gets it, almost like mitosis in human chromosomes. There are amazing systems out there. And the more we learn about the biology, it's like the more we understand how these are maintained, even before we were around to try and kill cells with antibiotics.
Yeah, very cool. All right. That's a great story. I really like that. Brings back memories. All right. That is your last TWiM of 2023. You can find the show notes at microbe.tv/TWiM if you like our work. For January anyway, go over to parasiteswithoutborders.com and make a donation. Otherwise, microbe.tv/contribute. And if you have any questions or comments, please send them to TWiM at microbe.tv. Petra Levin is at Washington University in St. Louis. Thank you, Petra.
Thank you very much.
Thanks for another year of TWiM. It's been a lot of fun. Looking forward to another one. I'm Vincent Racaniello. You can find me at microbe.tv. I'd like to thank the American Society for Microbiology for their support of TWiM and Ronald Jenkins for the music. This episode of TWiM was edited by Ray Ortega. Thanks for listening, everyone. We'll see you next year on This Week in Microbiology.